Hello, friends. Myself, Dr. Shalini Malhotra. Today, I will be talking about photophosphorylation. Photophosphorylation, as the name suggests, it is the production of ATP in the presence of light. Now, at the end of this lecture, you will be able to understand the process of photosynthesis, about the two photosystems, and about how the ATP is produced. Now, what is photophosphorylation? It is a process by which ATP and NADPH are produced using sunlight as a source of energy. Students, as you all know, the capture of solar energy by photosynthetic organisms and its conversion into chemical energy of reduced organic compounds is the ultimate source of all biological energy. Photosynthetic organisms and plants, they capture solar energy, they produce ATP, they produce NADPH, and they use ATP and NADPH to fix carbon dioxide into carbohydrates, and they release oxygen in this process. Yes, and they use carbon dioxide and water. And heterotrophic organisms, like for example humans, we use this oxygen to degrade the energy rich compounds, and in this process we produce carbon dioxide and water, and we synthesize ATP. We use this ATP for our biological processes. Unlike NADH, which is a major electron donor in oxidative phosphorylation, this water is a poor electron donor. Means it has a standard reduction potential of plus 0.82 volts, comparable to minus 3.32 volt. 32 volt for NADH. Photophosphorylation differs from oxidative phosphorylation in photophosphorylation differs from oxidative phosphorylation in requiring the input of energy in the form of sunlight, and thus it makes water a good electron donor. Now, you all know that green plants, they uh, they synthesize their own food, and the major cell organelle which is responsible for trapping the light and converting carbon dioxide and water into carbohydrates and oxygen is the chloroplast. It is the structure within the cells of plants and green algae that is the site of photosynthesis and the process by which light energy is converted into chemical energy. You can see in the diagram it is a double membraned organelle, and you can see the stacks of vesicles. These are the grana, and the single, they are the thylakoid, and the stroma is in the center. Stroma is the cytoplasm, and the main photosynthetic pigments or the chlorophyll molecules, they are present within the thylakoid lamellae. Now, chloroplasts, they contain green pigment called chlorophyll, which absorbs the light energy and converts it into chemical energy. Now, the accessory pigments and antenna molecules, they help in light absorption, and they collectively form light harvesting complex. These light harvesting complex, they capture the sunlight energy. When the light energy is captured, electrons, they are emitted, and then these electrons, they are accepted by the primary acceptor. You can see in the diagram there is a reaction center, pigment molecules, and there is a primary acceptor. Now, we have two photosystems. One is cyclic, other is non-cyclic. This photosystem one is a cyclic photosystem, that is PS1 or P700. It is a type of photophosphorylation which occurs within the lamellae of chloroplast. And this cycling means the electrons, they are released from the system P700, and they again come back to the reaction center P700. This is the cyclic. These electrons, like you can see in the image, the P700 is excited, it receives energy, it takes energy from sunlight, electron is emitted, and the electron, it passes through a series of carrier molecules like ferredoxin, plastoquinone, cytochrome. In this and in this electron flow, ATP is produced, and the electron, it again comes back to the reaction center P700. Therefore, it is known as cyclic photophosphorylation. uh, uh, Cyclic photophosphorylation is a light dependent reaction in which the in a high energy electrons they are released in a cyclic pathway from P700 to PS1. Photosystem 1, or it is also known as plastocyanin ferredoxin oxidoreductase, is one of the two photosystems in the photosynthetic light reactions of algae, plants, and cyanobacteria. Now this is the photosystem 2. It is P680 non-cyclic photosystem. The non-cyclic means the electrons they are released from the P680 but they do not come back. There is a electron hole in the reaction center, and that electron hole is filled by the photolysis of water. So this non-cyclic photophosphorylation is associated with the photolysis of water, and you can see in the figure that electrons they are released and they go through a series of carrier molecules, and in this ATP is produced and NADPH is also produced. And these two photosystems, photosystem 1 and photosystem 2, they are they act together, like you can see in this diagram. These photosystems, both the photosystems, they are linked together, and then they are able to form ATP and NADPH. This ATP and NADPH, they are required for the carbon dioxide fixation, means carbon dioxide is converted to, it is converted to carbohydrate starch. So you can see in the figure it is a Z scheme. You can see the photolysis of water, two water molecules, that is producing 4 electrons, 4 hydrogen four hydrogen ions and oxygen. So the oxygen which is coming, it is coming from the photolysis of water. You can see from the you can recall from the process of photosynthesis that carbon dioxide and water, it forms starch and oxygen. So oxygen is not coming from carbon dioxide, it is coming from the photolysis of water. Now these two photosystems, they are the part of light reaction. Now photosynthesis, it encompasses two reactions, light reaction and dark reaction. Light reaction means it is for the synthesis of ATP and NADPH, but dark reaction does not means that it is taking place in dark. It is just to differentiate between the light reaction, that is it is used for the production of ATP and NADPH, and these the products of light reaction, they will be utilized for the fixation of carbon dioxide into carbohydrates. So that takes place inside the stroma. It is the process wherein photosynthetic organisms like plants, they turn inorganic carbon into organic compounds like carbon dioxide. Carbon dioxide fixation, wherein carbon dioxide from the atmosphere is converted into carbohydrates, and it is in fact the first key step of the Calvin cycle. You can see in the figure, rubisco is the major enzyme which is taking this is ribulose bisphosphate carboxylase and oxygenase, means it can take both oxygen and carbon dioxide. If the concentration of carbon dioxide is more, it will take carbon dioxide and it will form 3 PGA. This was all for today. Thank you.